Everybody, welcome to the last session of the microenvironments theme for the HBBE's annual summit. It's my pleasure to introduce Dr. Angie Sherry, who's one of the VCF fellows for the HBBE, and she'll be presenting her work entitled Translocation of the Microbiome on Fungal and Fibre Highways in the Natural and Built Environment. So over to you, Angie.
Okay, thanks for the intro, Darren. So good afternoon, good morning, good evening to everyone. Yeah, so I'm Angie Sherry. I'm a VC fellow within the Hub for Biotechnology in the Built Environment, and I'm based at Northumbria University in the Department of Applied Sciences. So yeah, I work within the microbial environments theme. As Darren's mentioned, my background is in molecular microbiology. So I really use high throughput sequencing methods as my sort of tools of the trade to characterize microbes involved in pollutant degradation. So I've done a lot of work in anaerobic hydrocarbon biodegradation in my past. And today I'm going to talk about a study which really focuses on confocal laser scanning microscopy methods rather than the sort of identification of the microbiome through next-gen sequencing methods. So this study is a proof of concept study, and I'm working with Dr. Jane Scott, who is a Newcastle University Academic Track Fellow, also within the Hub. Jane's expertise is in materials and textile science, and this is a really interdisciplinary study where we're teaming the molecular microbiology and the movement of cells with textiles to look at the directional movement and dispersal of microbes on a range of natural and synthetic fibres to enhance hydrocarbon biodegradation. We're looking for potentially novel ways of processing waste in the environment using textiles and fibre highways, which will ultimately lead to the development of environmentally responsive textile systems composed of natural and sustainable materials to clean up sort of oil spills and oil pollution. This was funded during COVID last year through the Environmental Biotechnology Network. So it's really embedded within science, technology, and the arts, this study. So as we all know, there's a global legacy of hydrocarbon contaminated systems, and the study really focuses on tackling this issue. So there's just a few images of oil pooling against the Louisiana coastline along Barataria Bay, which was from the Deepwater Horizon oil spill. There's also often other sort of oil pollution and spills within the natural environment, both in terrestrial and aqueous systems in the oceans. So the study really focuses, or it was initially sort of determined from the concept of this fungal highways. So a fungal highway is basically bacteria actively using the liquid film surrounding the mycelium of a fungus for translocation. So on this image on the left-hand side, you can see that this white shadow is the liquid film around the fungal mycelium. And if I play this video on the right-hand side, you can see this motile strain of Pseudomonas putida translocating in this liquid biofilm around the fungal mycelium. So research into the movement of bacterial communities along these fungal highways has previously been shown to facilitate hydrocarbon bioremediation. So there's a few examples in the literature where we have evidence of bacteria using these fungal highways to degrade pollutants. So both examples have shown polycyclic aromatic hydrocarbon degrading bacteria using the fungal highway to degrade pollutants. So basically you have the fungi which are able to work their way through a soil system, and then the bacteria will travel on that fungal mycelium, and you'll get a dual attack on the hydrocarbon from both the fungi and from the bacteria. So this is really enhancing rates of hydrocarbon biodegradation within the natural environment. The second study, again, studied the movement of PAH degrading Pseudomonas putida along a fungus. And they showed that the bacterial movement became fourfold more effective and was clearly directed, pointing at a role of the mycelia for both the translocation of chemicals and microbes. So the first phase of this fungal highways and fibre highways research study was to look at the movement of bacteria in the presence of crude oil rather than PAHs. And what we can see is this was really sort of to develop the methods to then go on to look at whether bacteria can move on fibres as highways. So it was a reproduction of some of the previous work, but in the presence of crude oil rather than PAHs. And what you can see here in this video is that the bacteria, Pseudomonas putida, are actively moving around the mycelium within this oil-free system. If you look at the top of the video here, you'll see that the cells are inactive when there's no fungi present. So they're really relying on the fungi in this water layer in order to remain active. And if we look at the oil-amended system, again, we can see that the bacteria are moving actively around the fungal mycelium, and there's no diminished activity in the presence of the crude oil. Again, at the top of the slide here, you can see that the cells are quite dormant when there's no presence of the fungi. And this was really an expected result. This is a hydrocarbon degrading species of bacteria and fungi. So this was expected, and it was really just to help us sort of establish the methods within the lab on the confocal laser scanning microscope in order to move on with the research. So textile fibres have previously been used as oil sorbents. So sorbents are classified as natural and organic. They can absorb up to 20 times their weight in oil, but they have limited buoyancy. So that's not really an advantage in aqueous systems. There's synthetic fibres, which have been shown to absorb up to 70 times their weight in oil, but they're non-biodegradable, so that's not a very sustainable solution to this problem. And then there are natural organics, which have been used previously for oil spill cleanup. So we have milkweed fibre and cotton fibre, which were demonstrated to absorb significantly higher amounts of crude oil in comparison to synthetic fibres. And they've also looked at different types of fibres. So this second study showed that loose wool fibres, so raw untreated wool, showed much higher rates of oil absorption compared to recycled wool-based textiles, these needle-punched non-woven fabrics, which were constructed from recycled wool garments. In addition, in that study, waxes and grease, which were put onto the raw wool fibres, were shown to improve the hydrophobicity of the fibres, and that also increased oil absorption. Those studies were from sort of late 90s, early 2000s and the 70s, just to bring it up to date. There's also been more recent studies where they've used human hair as an oil spill sorbent and animal fur to help clean the oceans and polluted waters. So this study on the left-hand side used these hair-based absorbent mats in order to show biodegradation of medium chain, so medium molecular weight hydrocarbons by immobilizing a specific bacteria on the hair mats. And on the right here, there was an article in The Guardian just at the end of May this year, which went on to report that about 500 hair salons within the UK and Ireland have signed up to this Green Salon Collective, where basically they're collecting waste hair from salons across the UK and Ireland in order to put them inside these oil spill booms to mop up oil spills on the seashore. Usually 99% of hair cuttings from salons goes to landfill, so this is really contributing to a much more sort of circular economy by using these waste fibres. So the first part of determining whether fibres can be used as highways by bacteria for hydrocarbon biodegradation was to look at the microstructure of the fibres themselves. So you can see here the 5 different fibres that we were interested in testing in the study, and they all have very different microstructures. So there's more complex microstructures or morphologies. So the merino wool here has these scales, and there's much smoother fibres such as the silk. So just to focus on those 2, we were interested in the influence of this microstructure on whether the cells would adhere to the fibres. So you can see here that if we pipette this motile strain of Pseudomonas putida onto the wool fibres, we get adhesion of the cells both on the borders of these scales as well as in the middle of the scales. So there doesn't appear to be an influence of microstructure on adhesion to the wool fibres. And with the silk fibres, again, we can see that there are cells attached to the surface, and we want to eventually make this quantitative by doing some cell counting to determine cell adhesion to the different types of fibres within the study. So this is very sort of early preliminary data to show that we think that we have sort of confirmed this proof of concept study, that we have silk microfibers acting as a fibre highway. So if you remember earlier on in the slides, I suggested that the bacteria use this water layer around the fungal hyphae and mycelium in order to translocate around it. And what you can see in this fluorescence micrograph on the right-hand side is a water layer around a silk microfiber. And again, this motile species of Pseudomonas putida seems to have an affinity for this water layer around the silk fiber, and maybe using it as a fiber highway to translocate from one area to another. So of course, all of those images were at a molecular scale, and to progress this research, we have to think about scale. So Jane has been performing some experiments, bringing the design studio into the lab, where we're looking at different stitch lengths, different types of fibres, whether the fabric is loose or a more tighter structure, and different compositions of the fibres to sort of determine the effect on that oil wicking capacity if we were going to use this in real world applications. We've also been discussing the structure of different fibres. So on the top right hand side here, you can see a large surface area going into a tighter core. And we've been thinking about the fact that this could wick oil into a central core, where we could then have the mix of microbes in that central core to get really sort of intensive, aggressive degradation of the hydrocarbons within a core structure. On the bottom left-hand side, we have this environmentally responsive material which curls in response to moisture. So we're also sort of hoping to investigate further environmentally responsive textiles. And on the bottom right-hand side here, we have a mixture of the fungal mycelium with the textile structures on a much larger scale than sort of at the molecular level, which I showed earlier. So in summary, we've seen on the fungal highways research, Pseudomonas putida translocating on the fungus, and it wasn't inhibited in the presence of oil, which we sort of expected. So Pseudomonas putida do take the fungal highway in oil-amended medium, and in the presence of fresh oil directly spotted onto solid medium. With our fibre highways, Pseudomonas putea appears to adhere to the wool and silk fibres, and the microstructure doesn't appear to influence that. I didn't show this, but we have some evidence that the fibres form channels which influence and direct the flow of motile cells. And we have preliminary evidence of a microfiber highway on a silk microfiber. Scale should also be considered. There's this great design opportunity of working with textiles to explore microbial interactions for a range of different applications. So we're moving on now to study the translocation of mixed microbial communities on fungal and fibre highways. So we have these enrichment cultures on the go, which are enriching for hydrocarbon, degrading bacteria from beach sediments. And we're interested in looking at the bacterial fungal interactions, bacterial textile fungal interactions. We'd like to investigate further how less motile members of the mixed communities move around. So we've heard a little bit from Drac about how viruses may be using bacterial cells to move, and this is on the right hand side here. This is a recent paper showing a spore, a bacterial spore, sort of taking a hitchhiking on the flagella of a vegetative bacterial cell. So other less motile communities sort of hitching a ride or swimming, gliding, swarming. Yeah, interested in that going forward. We'd also like to quantify the cells, as I mentioned, and the rates of translocation in some of these videos that we're taking on the confocal as well. And obviously to determine hydrocarbon biodegradation rates to see whether we are getting this enhanced biodegradation when we've got more than one hydrocarbon degrading component of the consortia. There's many factors which affect interactions between microbes and textiles, and going forward we'd also like to look into that, some of these a bit further, and they're just listed there. So we think that this research from the natural environment really translates into built environment applications. So obviously there's lots of hydrocarbon waste within the built environment. So we'd like to go on to think about novel methods and biotechnologies for processing wastes in the built environment, using these multi-kingdom interactions with textiles. We'd like to explore translocation of microbes on formites, which again Jack mentioned earlier, which are these inanimate objects that when contaminated with or exposed to infectious agents can transfer the microbes to a new host. This leads into investigating health risks due to microbial dispersal via clothing, formites, home textiles. And there's definitely scope to also look at deterioration of textiles or fabrics, novel biomaterials, novel construction materials, which may be caused by these mixed microbial communities within the built environment. And it just remains for me to thank Jane, my co-I on the project, and eBina and the HBBE for funding. And thank you all for listening.
Thanks, Angie. So some lovely, pretty pictures there of things moving up and down. You mean these fungal highways? So I think we've probably got time for like one question maybe, and I think I'll grab that. What's driving this movement down these highways? Are they moving towards a chemical gradient, or is this just random movement?
Yeah, it's thought to be chemotaxis, Darren. There's a little bit of research that I had added in, but I didn't have time to see it. So on the apical tips of the fungi, you don't see very many bacterial cells, and that's not completely understood at the moment. It could be due to that it's immature and that it doesn't have these fungal exudates that are surrounding it. There's also something in the literature that the bacterial cells can contribute thiamine to the mycelium as they move further down the mycelium. So that sort of came up from the research that we've been doing and is possibly worth exploring in the future. But yeah, it's generally a nutrient or a chemotaxis gradient that the bacteria are sort of moving towards.
Okay, Angie, that's great. I think in the interest of time, thank you very much. Virtual clap again, and thanks very much. Another great talk. Thank you.